morning, good afternoon, or good evening. And I thank the committee very much for allowing me to present this work to this conference. Also want to thank the committee for forcing me to put on a bow tie. It's been a minute, and it quite frankly feels pretty good. So today I will be presenting work from my team at Roche Sequencing Solutions on a new tumor sampling method that we believe addresses the heterogeneity that we see in solid tumors. First, some acknowledgements. You can see the team on the left. It's a pretty small team. We have some very active and very fruitful collaborations with the Francis Crick Institute and the Royal Marsden Hospital, both in London. Specific callouts to Charles Swanton and Sam Turlich for their considerable help in trying to pioneer this new processing method that I'll show you in a few minutes. So my team is really interested in the diversity at the cellular, the genomic, and the transcriptomic level of solid tumors. One of the challenges we have, though, as a community of scientists, is that the way that we are currently sampling tumors leads to bias in the data that we get from the solid tumors that we're sampling. You can see this on the cartoon on the right. This shows 3 different biopsies taken from a tumor that has some variable areas of heterogeneity. And as you can see, each of those samples delivers different data sets, both on the genomic and then in a more general biomarker sense. So we believe that to really capture the diversity of solid tumors, we have to rethink how we're sampling in the pathology lab. And if you think about electoral polling or the mining industry, where the goal is to try and capture the most representative sample of a spatially segregated sample, the tool that they use is called representative sampling. And as you can see here, as we click through the animation, this is a very straightforward process that has been used, as I mentioned, in a couple of different industries. It's based on statistics that were defined back in the early 1950s by a mathematician who was in the mining industry named Pierre. And essentially what it says is that you take a spatially segregated sample, you homogenize it, and then every sample you take will contain the diversity of the original lot in every aliquot. And you can see that represented in this cartoon. So essentially, this is what we are attempting to do. And as I hope to convince you through this talk, in doing so, we generate some very interesting data, and we capture diversity that is lost in single biopsies. So to just touch briefly on how we currently sample solid tumors, I'm very aware that many pathologists are probably watching this and invited to this seminar series. So I'm not going to belabor the point on the left, where we have a couple of diagrams of the standard samples taken for the TNM sampling and staging. And here you can see a lung and a kidney tumor. And the samples are taken, many times are taken with the tumor and the normal tissue, such that you can look at the interface. For many reasons, these samples are critically important so that we can stage the tumor appropriately, which is both prognostic and very important when we discuss designing treatment regimens for patients. However, a single sample cannot capture the diversity of the entire tumor mass. This is very well demonstrated in the literature, both from Charles Swanton, Samra, and our work. The challenge that we have is that spatial bias, taking a single sample from one area, leaves you blind to biomarkers that are present throughout the rest of the tumor. This is also described as clonal illusion. On the right here, you can see data that is taken from a paper that we just recently published in Cell Reports, Litchfield et al. In the data that you see on the right, what we did is we did a survey of 76 samples of colorectal melanoma and sarcoma. And we looked at what is the average size of these tumors, and then what is the median percent of that tumor that made it into a PCR test in which 3 10-micron sections were taken for purification of DNA. And as you can see, if we just focus on the far right, the tumors themselves are about, on average, the size of a ping pong ball, whereas the amount of tumor that makes it into the PCR test is 0.0005%. And what this does is this generates the law of small numbers. The law of small numbers is yet another sort of cognitive bias where samples that are not representative tend to generate variable data, especially when we look at biomarkers or really any types of data that are present at low prevalence. So if you have something that is, say, 10% across a cohort of 1,000 patients, say, there are many, many times though you will just miss that and you will not understand really the total diversity in that study. And this is really one of the things that we believe this process is going to help with across the board. So this is a cartoon that demonstrates our workflow. On the top, you can see that the current tissue workflow for surgically resected solid tumors, the surgery happens, the tissue goes into formalin fixation, the samples for the TNM staging are taken, and those will be FFPE blocks. Those blocks, sections are cut, and those go through the standard diagnostic modalities that exist today in the clinic. A really important point here is that the sample that we are using, samples that we are homogenizing, consist of the residual tumor tissue that is not incorporated into FFPE blocks. We are not using FFPE blocks. We are using the stuff that gets put into the trash, and today is almost universally incinerated, with the notable exception of these samples being biobanked by a number of different companies and institutions. What we are doing is we are having surgical pathologists or pathologist assistants dissect out the rest of the tumor and a portion of normal, as you can see on the bottom. We then homogenize these samples, creating essentially a liquid tumor of that solid tumor. We then use the normal as the control when we sequence the tumor. And this has essentially been the basis of the work that went into the paper that I mentioned in Cell Reports. One point that I would like to point out again, just to repeat, we are not using FFPE blocks. Many times I do get questions about this, and we are only using the formalin-fixed tissue that remains after the TNM staging that goes back into the bucket and waits until sign-off. So here's just a brief presentation overview. Today I'll be talking briefly about some NGS downstream of representative sampling. We call this REP-Seq. This is sort of the foundation of the paper that I mentioned earlier. But then I want to get into some really interesting stuff where we've furthered our methodology such that we have enabled flow cytometry of intact cells from formalin-fixed tumors. And then I'll finish with how we're trying to use the flow cytometry and flow sorting to enrich for tumor cells prior to NGS, which significantly improves the data output. So first, we're going to talk a little bit about how representative sampling helps next-generation sequencing of solid tumors. I'm going to present just a brief case report. This is of one very large kidney tumor. There's a clear cell renal cell carcinoma. In this tumor, our colleagues at the Crick and at the Marsden sliced this tumor into 4 very large slices, otherwise known as bread loaves. From these slices, 60 punch biopsies were taken, and the remainder of the tissue was homogenized. I'll also be showing that blood samples were taken both pre-surgery and post-surgery, and we used many of the findings from the homogenate to better track the disease progression over time. So the first and most important aspect of representative sampling as it relates to NGS is the fact that when you homogenize tumors, you eliminate clonal illusion. As you can see in the top right, the little pie chart, this is essentially a brief description of what clonal illusion is. When you take a single sample, for instance, either the box that is across the purple and the gold slices of the pie, or the single box that's found within the green slice of pie, both of those samples are biased because they can't see the other parts of the pie. This is what is described as clonal illusion. Now, if you were to homogenize that, you would clearly see that you would, when you end up sampling from that, have about a third purple, a third gold, and a third green. And this is what we find in our case study. In this tumor, there were two major subclones. There was a subclone B and C that both derived from the clone A, it's the parental clone. And when we look at the single biopsies in the middle, what we see is that we have inaccurate clonality calls of the variants that we find in the individual biopsies, all of those 60 biopsies. So for instance, we have clonal variants from clone A that appear to be subclonal, but we also have variants in clones B and C that are subclonal that appear clonal. And just intuitively, when you think about subclonal drivers, subclonal resistance mechanisms, seeing that these things are clonal is not, number one, the truth for that tumor, and number two, may very well impact decisions downstream. Whereas on the right, when you homogenize that tumor and you sequence it following the homogenization, you get very accurate frequency calls. As you can see, clone A is vastly clonal, whereas clones B and C are all subclonal. And we've eliminated clonal illusion in this case. Some additional benefits when you use this method of REP-Seq for this patient. What we did in the middle box is we looked at, of those 60 individual biopsies, what was the reproducibility of the variant calls? And as you can see, the reproducibility is around 76% for any single biopsy. And what that means is any given biopsy is likely to miss 25% of the variants from any other biopsy. Whereas when you homogenize, you're around 97%. So it's almost totally reproducible. And this makes sense given the mathematical predictions of representative sampling. Another benefit on the right you can see is the number of predicted neoantigens that you can detect. From the single biopsies, on average, you're detecting just above 160. Whereas for the homogenate, we're detecting upwards of 180. But again, I think more important than the sheer number of neoantigens that might be predicted from the sequencing of these two different types of samples, it's really important to point out that each biopsy contains around 17% of those variants are thought to be clonal, when in actuality they are subclonal. And again, there's quite a bit of data out there suggesting that clonal neoantigens are the targets that will produce a very robust immune response, whereas subclonal and certainly very low prevalent neoantigens may very well not. And so our point here is that knowing the clonality and the appropriate clonality of a variant is really important when we start using next generation sequencing for downstream decisions for patients. So what we did with the initial exome sequencing that I've just showed you, apologies, I did mean to mention that all the data that you've seen so far are from exome sequencing. What we did is we then took all of the variants, it was around 76 variants that we detected in this patient from all samples, and we pulled them together to create a personalized targeted panel for this specific patient. We then turned that into a ctDNA test, and we sequenced DNA purified from serum that was taken before surgery, at surgery, and then post-surgery. And here you can see that each of the 3 clones are presented in their specific colors. And you can see that pre-surgery and at surgery, all 3 clones are represented at fairly high levels. Whereas right after surgery, 160 days after surgery, I should say, both clones A and B seem to have fallen off and are undetectable, whereas clone C predominates. However, that changes when the patient is on therapy and clones A and B take off, clone C is undetectable, and the patient then succumbs to their disease, and the predominant clones are clone A and P. It is interesting to note that in clone C is present at the autopsy in pleural fluid. But I think as I click through and you can see each of those variants presented as a blue box, we have P1, which is pre-surgery, P10 at surgery, and P16 post-surgery. You can see that what's interesting is the top boxes are predominantly the clonal variants, and many of those are not even detectable post-surgery, whereas they all come up at the autopsy. It's very, I think, interesting here to note that ctDNA analysis is rather challenging, and frankly, the more shots on goal that you have that are specific to that patient, the better outcome you're going to have. To that end, very important to note here that on day 160, the first time point after surgery, if we were to create a personalized panel from the single biopsies, 53% of those single biopsies would have missed tumor burden post-surgery at the first time point. And if you think about that, that would essentially be a minimal residual disease false negative. So I guess our point here is that not only is representative sampling very important when making decisions of those variants that are coming out of solid tumors, specifically around the clonality of those variants, when one then uses that sequencing data potentially to make a personalized ctDNA monitoring test, the more shots on goal, the better. And as we demonstrated for this patient, you will generally, and in all cases so far, get more shots on goal when you homogenize the tumor versus a single biopsy. So much of that data is presented in the paper that I mentioned that was published in Cell Reports, and I'm happy to point you in that direction. What I really am interested in, especially for this audience, is our ability to further process this sample and then enable it for flow cytometry. Interestingly, when you put a formalin-fixed solid tumor in a blender, it does not become what you think it would be. When we first started this, I kind of thought, well, when I make banana shakes at home, I no longer see the banana, so it must be liquefied. Whereas you can see on the image on the right, a formalin-fixed solid tumor gets chopped up into really small bits of tumor, including single cells. So we have chunks of tumor that are comprised of many tens of thousands of cells and single cells, but we have preserved the unit of heterogeneity, which is the single cell. We can then further disassociate those fragments of tumor into single cells using non-enzymatic methods and more mechanical disassociation along with filtration. And depending on the size of the tumor, we can create trillions upon trillions of cells, both cancer cells, immune cells, and stromal cells. I won't belabor this because I know that the vast majority of people in this audience are very familiar with flow cytometry, but just suffice it to say that what we are doing is, again, disassociating these solid tumors into single cells. This is non-enzymatic, so we're not degrading the epitopes on the surface of the cell, and then we are putting these cells through a flow cytometer. One important thing I think is of note is that, around 40 years ago, in the 80s, there were some papers where people disassociated tumors from formalin-fixed paraffin-embedded samples in isolated nuclei, which we have also done. However, the formalin fixation we find, really damages the cell membranes and creates very fragile cells that are really not amenable to flow cytometry, which is why I think we don't do today flow cytometry on formalin-fixed cells, at least of intact cells. And so I hope to convince you in the coming slides that this method is very robust. It's very similar to FRESH, and I think has some really interesting outputs that I believe the viewers of this seminar will really, really appreciate. So fixed flow, it's kind of what we're calling this. Fixed flow is very reproducible. As you can see in the cartoon on the left, we take a solid tumor, we homogenize it, we then disassociate it further and filtrate it so that we create a single cell suspension, and then we stain in 3 different experiments for the same biomarker, and then we look at the variance of that. As you can see on the figure on the right, this is the reproducibility of fixed flow. Looking at 2 biomarkers, CD3 and CD8, the coefficient of variance is incredibly low across 30 different patient cases. These tumor types include breast, colon, kidney, and lung. And if you look on the figure in the inset with the colorful dots, what we've done here is essentially looked at the biological relevance of the data, making sure that we're not wildly off, as CD8 is a component of CD3 positive cells. We certainly don't want to see a bunch of CD8 positive cells and very few CD3 positive cells. And what you can see here is that the data that we're collecting here looks very similar to what data should like when you look at CD8 and CD3. In some instances, we have more CD3 than we have CD8, as you can see the 2 blue dots on the bottom. But suffice it to say that the data that we're producing across 4 different tumor types is biologically reasonable, and as I'll show you in the next slide, pretty similar to what we get when we do that flow from fresh tissue as well. And this is that experiment. What we've done here is we've taken a biopsy, we've split it in half, and for one of those biopsies, we fixed it in formalin overnight and then disassociated it, versus the other biopsy that was kept fresh and followed a standard fresh flow protocol. As you can see in the middle, these are the images from the flow. You can see also very important to mention that when we do fresh, we do have to do live dead discrimination. And the percentage of cells that are dead varies across samples. But we subtract out the dead cells, we go with the fresh cells. Very important to note, but not too surprising. We don't do that in the fixed because all of the cells are dead. So when we look at these data on this from the same biopsy, they look very, very similar. And then when we look at these targets across all samples, all biopsies that we've done, most of them look very, very similar, almost identical, and the correlation is very, very tight. However, when you look at the figure on the far right, when you're looking at biomarkers for macrophages in the blue, these don't necessarily look similar. And I think I'll just mention a couple of things that we believe this might be the case. We're looking into this a bit further, but let me just describe what I think is happening here. It should be noted that the antibodies that we are using in this test are designed by Ventana for formalin-fixed paraffin-embedded tissue. They work very well on formalin-fixed cells when we use them for flow cytometry. That being said, it is likely that not all of these antibodies will work very well on fresh samples. So, you know, the antibodies that we're using to mark macrophages maybe just don't work great for fresh macrophages. So that's one possibility. The other possibility, which we're very interested in looking at, is whether or not, potentially, some of those macrophages are the dead cells in that live dead discrimination. One of the reasons why we think this could be the case, and as I mentioned, we're following up, is that, you know, one of the challenges with the workflow of fresh flow cytometry is that, you know, the hospital is in one location and the research might be done at yet a different location. And cells don't just hang out and are hunky-dory at 4 degrees in specific buffers, they change over time. And one of the benefits of fixed flow may very well be that we are taking a snapshot in time because the formalin freezes all of the biomarkers in cells essentially where they are. It also definitely helps with the workflow. You can fix these cells and wait weeks, as we do, until we disassociate them. Further, you can disassociate these fixed cells and then wait a few days before you do experiments. So we're very interested in how this workflow can change, flow cytometry and the analysis of cells from solid tumors. But suffice it to say that these data are very similar to FRESH, very reproducible, and allow us to do some really interesting analysis of these tumors, as I'll show you in the next slides. What can we do with this? Well, we believe that this allows us to really analyze the complexity of solid tumors in a fundamentally new way. Really what we can do is we can look at really any cell type across an entire solid tumor. And as you can see, we can look at any number of different cell types. So we can really very deeply interrogate the immune cells and the types of immune cells that have innervated that tumor over time. We can look at fibroblasts. We can look at the percentage of cancer cells. Also, we can look at endothelial cells. And what we can do is we can take the ecology of the solid tumor and we can describe it in its entirety across many, many, many different patients. And then we can compare these patients to each other, as you can see in this slide. Here are a handful of cases that have been processed from a clinical feasibility study run at the Royal Marsden with Sam Turlich as the PI. We are looking at melanoma, ovarian, kidney, sarcoma, breast. This is a pan-cancer study. And it's really looking at the feasibility of homogenizing solid tumors. As you can see, really these tumors, some of them look similar and some of them look very, very different. And this is really what we're finding across all different tumor types that we've looked at. What this flow cytometry of fixed tissue really enables us is to really better appreciate the diversity at the cellular level of tumor composition. Here are 3 lung and 3 colon cases that I do specifically want to call out, specifically these stage 2 tumors that you'll see both in the lung at the top and the colon at the bottom. So these are 2 tumors, same stage, but they are dramatically different in their cellular composition. For instance, LO2, just the designation that we've given, it only has 11% cancer cells, whereas LO7 has 36. The percent of cytotoxic T cells is also different. And so you can see that even tumors in the same stage look very, very different from each other. And frankly, LO2, that is a stage 2 tumor, looks very similar to LO9, which is the stage 3 tumor to the left. So we just find these differences to be very, very interesting. The same can be shown down in the bottom with the colon. Again, we have very different percentages of cancer cells, but very strikingly, we have in CO8, 13% cytotoxic T cells, whereas in CO6, we only have 4%. We haven't followed up on this, again, because all of these patients and this tissue is taken at surgery. All of this work is prospective. So we don't necessarily have deep follow-up and certainly not 5-year follow-up on any of these cases. But it's very interesting to think what the difference between 13% cytotoxic T cells and 4 in the same stage of the same tumor type might generate relative to response to immunotherapy. So I want to end the talk here by presenting some really interesting data that I think a lot of you might be very interested in, especially as many, I believe, in the audience are anatomical pathologists. What we're interested to do here is we are looking to enrich for the tumor population using flow sorting such that we are getting a more pure next generation sequencing library before we sequence these tumors, both saving sequencing reads and frankly saving money. So when we sequence bulk populations, we are spending quite a few sequencing reads on normal cells. And if we're interested in the variants from cancer, frankly, those reads are wasted. You can see if you look into the TCGA and many other repositories of sequencing data for solid tumors, purity of those tumors is a very significant challenge. And in many instances, very strict incoming criteria have to be made such that cases can be sequenced. And this is also true in CLIA labs and other labs doing clinical sequencing. You know, it's really important to have a specific percentage of those reads be cancer, because anything, say, under 20% is really going to be at risk when we're thinking about sequencing of solid tumors. On the bottom right here, you can see, again, two different tumors from this clinical study called Holstef, run out of the Marsden. The tumor on the left has around 25% cancer cells, whereas the tumor on the right has around 60% cancer cells. So those are two very different outcomes when we're thinking about sequencing. The tumor on the left is going to, we're going to have to sequence that to much higher depth to get the equivalent depth of the tumor on the right, which will potentially cost time and maybe even resequencing. So our goal here is to use the same methodology that enables flow cytometry of these fixed samples, and then sort these tumors to enrich for cancer cells prior to creating our NGS library. So you can see here in this series of figures the benefit of doing this relative to sequencing depth and output. In the bottom left, this is a colon tumor that was sorted based on cytokeratin 818. And you can see here that on the bottom left, 44% of that tumor was CK818 positive. We then sequenced both the homogenate, the mixed population of cells, and the enriched tumor. And you can see in the top right that the flow enriched sample, which is the cells that were sorted based on cytokeratin 818, had almost a doubling in tumor purity, even at the same sequencing depth. And so our tumor cell coverage is more than doubled, which then generates on the far right 65 additional variants that we find, versus in the homogenate, there were only 5 variants we did not find in the flow enriched, and those 5 variants hovered around 5%, which was our cutoff. More importantly, those 65 additional variants better enabled us to understand the complexity at the level of the phylogenetic tree of this specific case. So you can see in the homogenate, when we look at it, we only really see 2 subclones, very similar to the kidney tumor that I showed you as our original test case. Whereas when we sort this tumor, we see that there are 3 additional clones that weren't even found in the homogenate. Moreover, we see that one of those clones has what we describe as a leaf on it, so an even further subclone that is from that population on the far left in orange. So not only does enriching for cancer cells generate higher tumor cell coverage, but it also can enable us to better understand the evolutionary trajectory of solid tumors. So we have done this across samples from our clinical feasibility study. Here we have breast, ovarian, GIST, and kidney on the left. You can see that in all 4 cases, we consistently have enrichment from the homogenate. And on the right, every time we do enrich, we detect more variants. In some instances, we detect more than double the number of variants in the homogenate, and in other cases, we find around 20 to 30% more variants. Suffice it to say that these data are incredibly helpful when we're trying to look at what I mentioned earlier, which is the clonal solutions of these tumors relative to the potential evolutionary trajectories. So when we think about enrichment relative to clinical findings, I do want to point out one specific case, that one of the kidney cases that I just showed you. So in this case, we had 3 FFPE blocks, and when we do exome sequencing, what we see is that in FFPE block 1, we have a clonal VHL mutation, and we can see a low prevalence BAP1 mutation. However, in FFPE block number 2, we only see the VHL clonal mutation, and we miss the BAP1. And in FFPE3, the BAP1 G194A mutation appears to be clonal. That is clonal illusion, whereas it's actually subclonal. However, in the sorted REP-Seq, not only do we totally define that the VHL is a clonal variant, we better define the actual prevalence of the first BAP1 mutation, and we see a second subclonal BAP1 mutation. So we have convergence on this BAP1 mutation through multiple mechanisms, and we describe that as convergent evolution. And this is important, and if we look at this relative to the outcome of patients with these molecular features, and this is data taken from Samar-Turlich et al. It was a Cell paper back in 2018. A VHL-only mutation, those patients had a very, very positive outcome. Whereas when you have a BAP1 mutation that accompanies a clonal VHL mutation, those patients do not have as good of an outcome. And so really what we're showing here is that not only are we able to, with representative sampling, followed up by next generation sequencing, capable of defining the true mutant allele frequency of these very important clinical variants, we can also detect more clinical variants that do have very important outcomes relative to progression-free survival and overall survival. And in this case, it's very important to note that were we to have chosen FFPE block 2, we would have only seen the VHL clonal mutation, and that patient would have had a prognosis that was actually quite rosy, whereas in fact, they had a disease that would progress much faster. So I'd like to summarize this talk by, again, just presenting that representative sampling does capture the diversity at the genomic and the cellular level of solid tumors in a mathematically powered way versus single samples. It's important to note here that I think many of the people watching this seminar will understand the sheer amount of solid tumor tissue that is incinerated. There are hundreds of thousands of patients where this is likely happening. And I hope that you're at least a little convinced that that data that's in that tissue could be very important when considering how is the diversity of the ecology and the diversity of the genomics of cancer patients playing a role in outcome. And I think that if we, as a community, reassess using a very statistically standard methodology of surveying and sampling, it's used in electoral polling in the mining industry, we can begin extracting those data sets and better powering our studies to truly understand how heterogeneity is playing a role in solid tumor biology and progression. We can further disassociate these homogenates into single cells, thereby enabling whole cell flow cytometry. Further, we can then sort those samples and then go ask questions about specific cell populations that we're very interested in. Data I showed you was we can sort and enrich for cancer cells, thereby improving both the metrics around next generation sequencing, like tumor cell coverage, and the ability to detect clinically relevant variants. So with that, I will end this presentation. Again, I would very much like to thank the organizers for inviting me. This is the first time I've actually put on a long sleeve shirt and a bow tie since March. I thank everybody for allowing me to have that opportunity, and I hope you appreciated the talk. So thank you very much.